What's going on, everybody? Today we're going to be talking about the cell cycle and basic cell replication. So here's the basics for cell cycle. We start with, we have actually two phases, and then we have some subphases. So we have interphase, which is, the cell doing life, and then we have mitosis or meiosis, which is the cell actually splitting and making two daughter cells, or in meiosis, four daughter cells. So in interphase, we have G1, S phase, and G2. We'll get to that later. And then we have mitosis, which is your somatic cells. Somatic means yours, stomach cells, liver cells, skin cells, any cell that's not a sex cell. Sex cells are meiosis, and those are two different processes which are a little different. And in prokaryotic cells like bacteria, you're going to see binary fission. I have a little bit on that. You don't really need to know that for any high school level class. So some vocab that we should get out of the way. Haploid versus diploid. What did my one biology teacher used to say, or my only biology teacher used to say? I call it diploid. Some people call it I don't even know how you would say that any different, but if you're watching this, please pronounce it for me again, because I have no idea. So haploid is one set of chromosomes. Diploid is two sets of chromosomes. You'll see another synonymous term is homologous and non-homologous. Haploid being non-homologous. Homo meaning same. So same chromosome, two pairs. So homologous and non-homologous. Homologous being diploid, non-homologous being haploid. So let's start in the general cell cycle. This is going to be meiosis and mitosis. We have G1. This is the cell growing and doing its cell functions. This is going to be the longest part of the cell cycle. So think about in your human life. You have your baby years, your teen years, your adult years, and then your grandparent years. G1 would basically be your adult years. You are growing, or that's kind of stupid, but you're spending the most time there. That's what I'm trying to get at. You have S phase, where you're duplicating the DNA. S stands for synthesis. So when you think S phase, think synthesis, to build. So the reason we actually do DNA replication is so both daughter cells have the correct amount of DNA. If you think about it, if we were to just split right now without S phase, we would have a cell with half the amount of DNA that it needs. And if you know anything about cells, you lose half of your brain. Think about it. If someone just took your brain, cut it in half, and you lost one side of your brain, oh no, you're not. I don't even think you'd freaking live anymore if you think about it. So, Next we have G2. This is just going to be prepping for division. You don't really need to know a lot here. It's just a checklist, really. We're going to say, okay, do we have everything we need before we split? So before we get to mitosis, we need to understand what phases there are. We follow PMAT, and then we have a C at the end. You'll hear PMAT a lot, but I like to say PMAT-C. It stands for prophase, metaphase, anaphase, telophase, and then cytokinesis. So prophase, the chromatin, which is your uncoiled DNA, is going to condense into chromosomes. If you guys don't know anything about chromosomes, chromatin, I'll leave a video for that. I will most likely make a video on that sometime in the future. And then the nucleus will disappear. And then we have metaphase, and the chromosomes are going to line up at the equator. We'll also hear the metaphase plate is what they like to call it. And then the centrioles are going to attach those little spindle fibers, and that's what's going to pull them apart in anaphase. And anaphase is going to pull the chromatids apart. Chromatids are one part of the chromosome, don't forget. It's going to pull it to opposite ends of, that should say the cell, but it says chromosome, I wish I would have done that a little different, but we make mistakes. We're all human here. And then we have telophase. The nucleus is going to reform, and then the chromatids are going to uncoil into chromatin. Don't get those two words confused, chromatid, chromatin. Chromatin's more like the spaghetti-like DNA, and then chromatids are like one side of a chromosome. So please, for the love of God and everything that is good, do not mix those two up, because it's not fun. And then we have cytokinesis. This is a very interesting thing that I find actually extremely fascinating. How a cell can really just do this. It's just so odd to me. So In mitosis, we're going to have two diploid daughter cells at the end. We'll see in meiosis it's a little bit different. But in animal cells, we're going to see the cleavage furrow, which is this right here, this little thing. I don't like how they use the word cleavage. I think that's a very interesting choice of vocabulary. But if you think about it, cleavage, like, of course, you've probably heard it a million times. I mean, nobody wants to say it, but it's true. We hear that, like, in places where we shouldn't. So, you know, it's kind of like that. It's going to, like, pinch. It's going to pinch that, and then we're going to have two separate independent animal cells that are diploids. So, and then the same thing for plant cells, although they don't actually become independent from each other. They become independent organisms, but they just form a cell plate. So if you think about it, it's kind of like they're just welded together basically, but they just separate by that cell wall. It is so weird and confusing how they just are like, if you think about it, like at the end of the day, they're kind of just one big cell. But they're not at the same time. It is so weird to me. But, yeah. That's it for cytokinesis. Now we have meiosis. So something we should know, I probably should have put a slide for this. Meiosis does PMAT twice, PMAT-C twice. This is so we get 4 haploid daughter cells. We'll get to that a little later. But we have prophase 1. Same thing as last time. The nuclear membrane leaves, chromosomes are going to actually cross over here. What is crossing over? It's confusing at first, but, you know, just keep it in your brain how this works. So unlike prophase 1, where it's just the nucleus leaving, here what happens is the chromosomes, they're going to be homologous pairs of chromosomes because in meiosis we have more chromosomes. Unlike mitosis, I probably should have put a slide for this, but there's more chromosomes, and what they're going to do is they are going to line up with each other. They're going to line, like, butt up next to each other, and they're going to cross over DNA. And if you can see in this little graphic down here, we have the homologous chromosomes. They match up. You know, you get one from mom, one from dad. They swap DNA. And this is actually how you get your genetic variation. Like, you look at your brother, and you're like, you look nothing like me. That's because of crossing over. So be happy for that. And then we have metaphase, same thing as last time, but here is where it's important. Remember in metaphase, in mitosis, we had 1 line of chromosomes. Here we have 2 lines of chromosomes. So keep that in mind. And again, they line up at the equator to the metaphase plate. The spindle fibers connect to the centromere. I don't think I actually explained this. The centromere is that little middle part of the chromosome. And in anaphase, they're going to pull those homologous chromosomes apart. So we go from homologous chromosomes to 1 pair of chromosomes. Telophase, same thing. And then cytokinesis will occur, and then we will have 2 independent cells. So we kind of get back to mitosis here, but it's a little different. It's a little different because we're going to make haploid cells instead of diploid cells. So like prophase in mitosis, we have prophase 2. The nucleus will disintegrate. We have those 2 daughter cells that we made from PMAT-C1. Now we're on PMAT-C2. We have metaphase. They line up. The chromosomes line up at the metaphase plate. Spindle fibers connect to the centromere of those chromosomes. But unlike mitosis, where we had the chromosomes splitting up, we have the chromatids, or we did have the chromatids in mitosis. See, I always confuse myself with this, guys. It's confusing. But the centrioles, they pull apart the chromatids to opposite ends of the cell. And then we get 4 different types of cells. And remember, we had those homologous chromosomes. Now we have just those single chromatids. We have 2 chromatids, so it's pretty crazy to me. And they make 4 haploid cells. Boom, bam, pop, and that's it. You have 4 new daughter cells, haploid. Remember, 1 set of chromosomes in that cell. And why are they haploid? You might say. You mean like it's like, well, they need both. They need to be diploid if they want to be actual cells. Well, think about it. Your sperm and the egg meet, and they actually connect the DNA together, and that's how you get you. Those 2 cells make a zygote, I think they're called, a fertilized zygote. I think zygote is literally just a fertilized egg in definition, but you know, the 2 meet, and you get you. So pretty awesome if you think about it. You get all your chromosomes at least. I'd hope so. And then you have binary fission. This is actually really interesting. I thought I'd just throw this in here because it's kind of relevant. Prokaryotes don't really have the cell cycle, you could say. They kind of just replicate the DNA. They're going to do this thing. They're going to elongate, and that DNA is going to separate to opposite ends of the cell, and they're just going to split. So they do replicate, split, replicate, elongate, split. It's so interesting. I definitely recommend that you do some, if you guys are really interested in the topic of biology, that you go and actually further your knowledge in this, because I went down one big damn rabbit hole on this, and it was interesting to say the least. This is kind of just the basics of it, but it is really interesting, and I feel like you guys would learn a lot. So I highly recommend you check out some other videos. I think they explain some of the chromosome numbers better than I do. I actually think I'll make a chromosome video number. Like, it is so confused. Like, even I still do not get the chromosome numbers. Like, I try to make graphics all the time. It just doesn't work, guys. But, you know, it's just how it is. Well, that's going to be the end of this video, guys. If you guys did enjoy, please like, subscribe. Anything really would be nice, and I'll thank you guys a lot for that. Anyways, have a good one, guys. Bye-bye.